I'm really excited to present Antonio Pardiñas, that's the right pronunciation, for everybody, who comes from, this is a senior lecturer at Cardiff, so the go-to center for the genetics and the genomics in UK and worldwide indeed. He has a spectacular research career. Started with something that looks interesting, like a more anthropological research, but moving into the really cool things about schizophrenia and pharmacogenetics of this lovely drug called Clozapine. I'm not the important one. Antonio's here. The talk is spectacular, so go for it. Thank you.
Thank you very much, Emilio. I always appreciate your enthusiasm. I don't know if I'm fully deserving of it, but I'm very happy about it. Thank you everyone for having me here, for inviting me to give this presentation to such a select group in the UK. And as Emilio has said, I come from the Center for Neuropsychiatric Genetics and Genomics at Cardiff University, and I'm going to speak to you about the latest genome-wide pharmacogenomic studies that have been done for the antipsychotic clozapine. So first thing I want to say is that I don't have any conflicts of interest to disclose in relation to this presentation. And I do like to set the stage for this talk by putting this quote from a few years ago from a Lancet Psychiatry paper, which described some of the challenges of prescribing medications for psychiatric conditions. And even if just to say that what myself and many others who work in antipsychotic pharmacogenomics, in clozapine and in many other drugs, I trying to do is to shift this balance between art and science for prescribing practices as much as we can towards the science. And I know I don't have to speak to this audience about what treatment resistance in schizophrenia is, but also to set the stage. It's estimated that about 30% of people have symptoms that do not respond to 2 or more trials of antipsychotic medications. And this is the presentation called as treatment resistance, which increases the risk of a lot of bad treatment outcomes, decreases life expectancy, and severely increases the cost of care for these individuals. And of course, the pressures on them, their families, and carers. So this it's not strange to know that this is a research priority for patient organizations and representative groups, and it's the top research priority for schizophrenia as reflected by the James Lind Alliance. And of all of the challenges and of all of the complexities of treatment resistance in schizophrenia, we do have one drug, one medication that is evidence-based, and is the only medication licensed specifically for treatment-resistant schizophrenia. The antipsychotic clozapine is generally effective, is as effective for treatment-resistant schizophrenia as other antipsychotics are for treatment-responsive schizophrenia, and it is associated with high adherence to treatment and to a reduced risk of all of those bad outcomes that I had highlighted in my previous slide. And by the way, I forgot to say, but on the bottom of most of my slides, you'll find these letters PMID, and you'll find some numbers there. If you're interested or getting more context about what I'm speaking about on each slide, you just need to go to PubMed, put those numbers there, and it will give you relevant papers that I think are relevant. So just to say that. So the good things about clozapine are those, but clozapine has a very broad adverse effect profile as well. And the adverse drug reactions that most people are familiar with are the hematological type of adverse drug reactions, such as granulocytosis, which is idiosyncratic, it's rare, and is potentially fatal, and it has led to the blood monitoring system that is mandatory for clozapine, for people that go on clozapine treatment. But there's also another range of adverse drug reactions that all of which can be impairing, and if they're not properly managed, they can also be fatal, such as paralytic ileus, seizures, or weight gain. These in particular that I cite here are known to be dose-dependent reactions, and this is gonna be of some importance for what I'm gonna speak about. But a big problem with clozapine is that it's widely under-prescribed. In nearly all of the countries where people have done studies on clozapine prescriptions, it's been found that fewer people than those that should get it. And in fact, in the UK, it's estimated that only a third of those eligible for clozapine actually get the drug. And this is even worse in the United States, where over a certain period of time, I think a decade, amounted to about 5% of antipsychotic prescriptions. Reasons for anti-prescription are, of course, not easy to ascertain. Things that are argued in data to relate to, for example, insufficient clinician training. And coming from a medical school, I also have personal perspectives with this, when usually medical students get much more focused into this broad adverse effect profile than in the good beneficial effects, or the fact that it's the only licensed medication for treatment-resistant schizophrenia. But that's a reality, and indeed concerns about adverse effects are not only voiced by clinicians, they're voiced by patients as well. So with the research that we're doing, a question that always, or a name that came into us was, are we able to address this problem of anti-prescription by, as far as possible, eliminating some of the causes of adverse effects of the drug? And this brings us into a paradigm, which I imagine many of you will be familiar with, which is the paradigm of therapeutic drug monitoring. And this is used for many drugs, but it can be particularly useful for some drugs used in psychiatry. It's the measurement of the plasma concentration of a drug at designated intervals throughout time. And it allows optimizing the dose to achieve an optimal concentration, often called the therapeutic range. So the idea is that concentrations of a drug in the therapeutic range have a good balance between response and toxicity or adverse effects. When the concentrations get too high, the risk of adverse effects increase. When the concentrations are too low, the effect is likely to be subtherapeutic, so there's going to be very little response. Therapeutic drug monitoring is a great framework. It works, and is most useful when the drugs have a narrow therapeutic range. So there's only a small interval of plasma concentrations of the drug that have this relationship between potential benefits and potential adverse effects. And this is the case for many psychiatric drugs, and definitely is the case for clozapine. But when therapeutic drug monitoring cannot be used, and this definitely does happen in many countries where it's too expensive to put everyone through TDM, then we need to have some predictors of drug metabolism that can help us establish dosing guidelines. And common predictors of drug metabolism are gender, age, body weight, smoking habits, for example, is very important, and there are other things. But it's also known that for clozapine and for many other psychiatric drugs, about 50% of the population variability in metabolism is not explained by any of these factors. If it's not explained by any of these factors, how is it explained? What can explain it? The role of ascertaining what can explain it is the role, among other disciplines, of pharmacogenomics. And pharmacogenomics studies the genetic basis of pharmacokinetic processes and pharmacodynamic processes, with pharmacokinetic processes being what your body does to the drug, so absorption, distribution, metabolism, and excretion, and pharmacodynamics being what the drug does to your body. So anything related, in this case, for example, with antipsychotic effects. Okay. I'm going to go a little bit into the terminology of pharmacogenomics, even though these concepts are not going to come very often during the talk, but because they are the first thing that people find when they go into the pharmacogenomics literature. Pharmacogenomics has a very old-fashioned way of calling the genetic variants that are relevant. They call them star alleles or classic alleles. And what is a pharmacogenomic star allele in many other genomic disciplines is what we would call a haplotype. So star allele is an inherited combination of genetic variants, although some star alleles can be defined by just one genetic variant. And the importance of star alleles for pharmacogenomics is because star alleles are usually mapped to an assessment of protein function. They usually have a known effect on an enzyme or a protein, and that effect is usually called the activity value or activity score. The way that in a pharmacogenomics framework you go from the genetic variation to the activity score is depicted there in this slide. You have this information on the genetic variants, so SNPs, single nucleotide polymorphisms, that allows you to call the star allele, which then will have a potential function that maps to a value. And when you look at the potential combination of one star allele with the other star allele, remember that we have 2 copies of each of these, then you get the diplotype. The diplotype will map to a series of activity values. The sum of those will be the activity score, and that will give you the phenotype. And there are standardized terms to define these phenotypes that are here. Essentially, when you're talking about an enzyme that metabolizes a drug, the majority of people will be in the category that is called normal metabolizers, and people who are not normal metabolizers can be rapid metabolizers if they have fast metabolism, or poor metabolizers if they have a slow metabolism. So how can a framework that allows us to ascertain the genetic contributions to metabolism be of help in the clinic? Well, because atypical metabolizers, and this is usually 20%, it could be less, but it could be 20% or less of the population, they're more likely to be dosed suboptimally. And when those suboptimal doses lead to low effectiveness of the drug or to high risk of adverse reaction, then knowing the genetic variation that is responsible for those atypical metabolizers allows us to go into this framework of genotype-guided dosing, of using the genetic information to select doses that are more optimal for those subsets of individuals. And what you can see here are the results from a study on the antidepressant escitalopram, and I'm only going to briefly go over them, but here in the picture on the left, what you have is the different distributions of escitalopram concentrations in different groups of people that have been characterized by their star alleles. Okay? And you go from null, the people with the null diplotype, which have very high concentrations of escitalopram, to the people with the star 17 diplotype, which have lower concentrations of escitalopram. And the reason why this is relevant can be seen in the graph on the right, which is a graph with 3 axes. It's not what I would recommend my students to plot. It makes it complicated to explain. But if you just focus on the red line, what you can see here is that different groups of individuals, again, solely characterized by the genetic variation on one enzyme, by these star alleles, have different proportions of people who abandon treatment. So who go after trialing escitalopram, they change to a different medication. So escitalopram is not working for them. And that proportion of individuals who abandon the treatment or who discontinue the treatment are very high in the extremes, in the people that are atypical metabolizers, and are much lower in the normal metabolizers. Why? Because people that have high concentrations of escitalopram, much higher concentrations than you would expect, are more likely to get adverse drug reactions. People who have much lower concentrations than you would expect are less likely to get therapeutic effects. That's the general principle. And in order to see how this could work for clozapine, then just going to have a very quick look at how clozapine metabolism looks like. So clozapine is rapidly absorbed after oral administration in just 1 to 2 hours, and it goes straight through an hepatic pathway for first pass and secondary metabolism. It's the process, even though you have here a lot of arrows and a lot of enzymes there, is relatively simple. 70 to 90% of the drug is converted to N-dimethylclozapine, also called norclozapine. And this process is mainly driven by an enzyme called CYP1A2. There is secondary metabolism of clozapine, so everything that is not converted to norclozapine can go to another compound called clozapine N-oxide. And all of these compounds, including clozapine itself, can be excreted after a process called glucuronidation. And metabolism to clozapine N oxide is mainly driven by CYP3A4, and glucuronidation is driven by many enzymes in the UGT superfamily. So there's not that many genes implicated here, as you can see, but also all of these processes are potentially important, because even though clozapine is the only compound that is thought to have antipsychotic activity, and I say thought because there's not a lot of hard data on this, the source of adverse reactions is not really known. And all of the compounds that you have in this pathway here, and by the way, I didn't say that the pathway is derived from the FIME-DKB database, all of the compound that you have in this pathway here can be reactive under certain conditions, so could be the source or a partial source of toxic effects. We just don't know. So this is the biological picture of clozapine metabolism. But the clinical picture of clozapine metabolism is fairly important. And as all of you that are familiar with clozapine would know, there are many compounds that have drug interactions with clozapine. And those drug interactions occur because the co-administration of those compounds might increase or decrease clozapine plasma concentrations to levels that are not normal for the individual. They're not what the individual would have if they weren't co-medicated with those compounds. So it is important because that can have large effects on adverse reactions and on efficacy. And also there are adverse reactions such as seizures that are very well mapped to having high plasma concentrations. And when seizures happen, then a lot of the guidelines recommend doing actions to reduce clozapine plasma concentrations. So speaking of guidelines, I just want to highlight everyone to this recent update of 2022 of international clozapine management guidelines. I think some people here, definitely Emilio, was a part of doing these guidelines. And there's actually a paper that came out very shortly afterwards, which I think gives a nice complementary view. So I'm going to recommend these two papers. And what the guidelines establish very clearly is that the therapeutic range of clozapine is there and is narrow and is established in 350 to 600 nanograms per milliliter. And certain groups of the population have certain metabolic characteristics. East Asian individuals, or individuals of East Asian ancestry, require 60% lower doses to hit the therapeutic range because there is low metabolizers. Smokers are rapid metabolizers and require 30 to 40% higher doses to hit that range. So with knowing this, therapeutic drug monitoring is still useful for the management of day-to-day individuals, but there are added benefits to knowing this and adjusting the starting doses before therapeutic drug monitoring is even initiated. So knowing all of this is found as being fairly important. But something that the guidelines also mention, even though they don't devote a lot of space to it, is that there are no evidence-based pharmacogenetic variants for clozapine that can help in any way to optimize clozapine treatment, and that is as of 2022. Why is this case? Well, we can go a little bit further back in the literature and look at systematic reviews that were focused on genetic variants for clozapine, and there's lots of these. I just highlighted 4 here that I think are representative of the wider literature and that probably review nearly everything that is to be reviewed. And what these studies highlight is that the majority of studies that have been carried out in clozapine, and specifically for looking at genetic determinants of clozapine metabolism, were very small. We all have some awareness of how difficult it is to recruit individuals for research studies and how difficult it is to fund research studies in psychiatry. So it's probably not surprising, but there is a lot of studies that have been published that don't even have 100 individuals, and we know how statistical power is affected for that. Also, the majority of studies looked only at very specific genes, so they weren't even in a position to say whether said whether all of the genes implicated in clozapine metabolism had these effects or those effects, because they didn't look at all of those genes even, and you know, not to speak about the whole genome. And not only there was a limited number of genes looked at, but very few polymorphisms have even been looked at. And the majority of those polymorphisms were coding polymorphisms, which also might have some importance on what was found here. So the summary of all of these studies is that there were no robust results, at least reviewed during this time, on genetic variants with effects on clozapine metabolism, with maybe the exception of the ABCB1 gene, which is a drug transporter, and I can speak a little bit later about that. So that in itself, that situation when there were no robust findings, is not of any worry. It's very typical of research fields that are in its inception and consolidation, but it can get problematic when some results for a specific research are then taken through the clinic without due process. And this is a very interesting paper from 2017 describing a case study, arguably, it's only a case, about how the guidance for from a pharmacogenomic test was misleading because it predicted low clozapine response on an individual that then responded to clozapine. Of course, those type of situations are always going to happen. We can all think that, we can all imagine that with response to clozapine being highly variable, predicting it is going to be highly problematic in itself, and it's going to be something that sometimes is going to work and sometimes it's not going to work, even if we have the best possible predictors. But the interesting bit about this study is that the pharmacogenetic test that was used was looking for a single nucleotide polymorphism in the gene DRD2 and used that polymorphism to call poor response to clozapine. Now, the DRD2 is the gene that is the target of all antipsychotic medications that is known. But there is no study ever that has identified a link between DRD2 genetic variants and response to clozapine. So how this extrapolation was made, or how this association was made and then introduced in a genetic test, in a pharmacogenetic test, is not clear. But it's interesting to know that this at least happened once, and we all need to take or precautions for these type of things not to happen again. So, in order to be able to do that, then we're not alone and only in company of our own judgment. There are publicly available pharmacogenomic guidelines out there for many drugs, but they are at the moment non-available for clozapine. These pharmacogenomic guidelines are collected by the website PharmGKB. So if you're interested in this, I recommend you to go to PharmGKB and just look for your favorite drug and see what it says, what the expert reviews international consortia have said. And what there are guidelines for 6 other antipsychotics, and they're all based on CYP2D6 metabolizer status. Curiously, there is a warning in the FDA-approved drug label for clozapine about the potential of reducing the dose in patients who are CYP2D6 poor metabolizer. But where that primary data, study, or trial, how that was conducted is not unclear. Of course, it's not cited in the drug label. That's not a place to cite it. But I'm not aware of any reference that has given high data on that. So it's also called into question. PharmGKB does highlight 46 genetic variants that might have effects on clozapine, particularly on metabolism, but they all are classed as low quality of evidence. This is not a judgment about the study. It's not a judgment whether the study was better or worse that discovered those variants. It's just hints that those variants have been only shown by a single study, or they have been replicated by another study, but the replication is inconclusive. So it's a hard line that a genetic variant needs to have in order to be put into a pharmacogenomic guideline. but I think that's for the better. These guidelines tend to be heavily conservative. So now I'm going to go into how we can go about finding these pharmacogenomic variants using the study design called the Genome-wide Association Study, or GWAS. And for those of you who might not be familiar with GWAS, a very brief explanation is basically very similar to any other genetic association study. You need to collect data from as many people as you can. Usually GWASs collect data on the thousands of individuals, but of course, sample sizes can grow through collaboration and merging individuals from different collections. Then all of those individuals are genotyped in an array, and that array usually types about 500,000 genetic variants, although some arrays type more and some can type less. And quality control is used when you have that data from arrays in order to determine and control for potential study confounders that are very common in genetic studies. Interestingly, the quality control that we do in GWAS is quality control that you can only do when you have array data, when you have data on many hundreds of thousands of polymorphisms. So many classical genetic studies and candidate gene studies weren't able to conduct that quality control because they were based on the sequencing, for example, of a single polymorphism. And it has been retrospectively found that a lot of those studies were confounded because that quality control couldn't be even applied, because the data wasn't proper for that. So that's an advantage of genotyping arrays and an advantage of GWAS from the outset. Also, having those many hundreds of thousands of polymorphisms allow us to conduct a process called imputation, which what it does is it uses a reference data set to get information from genetic markers that are not in your array. And that means that when you look at the GWAS studies, you usually see that they have results probably for 5 million polymorphisms or more. Most of those polymorphisms have been inferred from the 500,000 polymorphisms that were in the array. But that inference process is very, very reliable and is now fairly well established as a gold standard for GWAS. And after doing that process of imputation and getting that data on 5 million markers or more, then we conduct association testing. And here we have the stereotypical data visualization tool for GWASs, which is called the Manhattan plot. And the only thing that you have to remember about the Manhattan plot, if you're not familiar with it, is that each dot that you can find here, see these little dots, each dot is a genetic marker, is a single nucleotide polymorphism, and the higher the dot is on the Manhattan plot, the better, because the height of the Manhattan plot is proportional to the statistical significance of the association. And usually there will be a red line, and everything above that red line passes the statistical significance cutoff and is considered a genome-wide significant variant. That's how we call it. Okay. So that's how GWAS is conducted. From the results of the GWAS, we usually conduct another type of analysis called the polygenic score or polygenic risk score analysis. And this is fairly simple as well. We have the data for the GWAS that tells us from each SNP what is the inferred effect. And we have then genotype data on a completely independent set of individuals. It needs to be completely independent because otherwise bad things happen. But say we have data on a completely independent set of individuals. So these are individuals that were not used in the GWAS. We have the genetic information at the same SNPs that were typed in the GWAS. And we only need to map this genetic information to the effects. This is, again, similar to what you could do if you were doing certain types of survey designs or certain types of testing for individuals in other matters. You have their information and you use this information to map it to a value. And that value is the polygenic score or the polygenic risk score. And a nice property of polygenic scores is that they tend to be normally distributed variables, so they are handy for statistical testing. So a lot of the research that we have done on Clozapine and that we have done on GWAS is powered by a dataset that we have in Cardiff University, which is called the CLOZUK project. And the CLOZUK project was initiated and is maintained by these three gentlemen there, Professor James Walters, Professor Michael Owen, and Professor Michael O'Donovan. And this dataset is based on anonymized samples that have been collected through Clozapine monitoring systems from all over the UK, thanks to the collaboration of the pharmaceutical companies managing these systems. It is at the moment the largest treatment-resistant schizophrenia dataset in the world that is linked to genomic data. We have 3 waves of the study at the moment, with in combination a total of more than 15,000 individuals, all of them with array data, many of them with sequencing data as well. And from a subset of it, we also have blood sample data, including full blood counts and Clozapine plasma concentrations. A big limitation of CLOZUK, and it's the one that we always need to be very careful when we discuss this project, is that we have no clinical or demographic information from these individuals. We know they were taking Clozapine. We are currently linking to NHS records, again, all over the UK, but this is a process that is taking time. And I just want to highlight, because particularly James likes me to put this figure about when I speak about CLOZUK, that the study is so large that when we do even very basic genomic analysis, we can distinguish the population structure of the UK just on the samples of CLOZUK. And this is the plot that you can see here at the bottom, where you can see that the 4 countries of the UK are fairly differentiated. This plot is generated solely from the genetic data. And as a population geneticist, I can tell you, if we had a smaller sample, we wouldn't be able to see this. And as James likes me to say, here you can see the Welsh people ascending to heaven, as is their rightful place. So what did we get when we had a first look at doing a GWAS for the Clozapine concentration data that we have in CLOZUK? We did this in 2019. We did a GWAS with about 10,000 Clozapine assays, the data from 10,000 Clozapine assays from under 3,000 individuals, all of which of European genetic ancestry. And we conducted in reality 3 GWASs. 1 GWAS for the Clozapine levels, 1 GWAS for the non-Clozapine levels, and 1 GWAS for the ratio of both Clozapine and non-Clozapine. And what we found was 5 genome-wide significant loci in total. For Clozapine, we found a single SNP, which is placed between the genes CYP1A1 and CYP1A2. This is because the genes are very close to each other, but it was a single SNP. Then for non-Clozapine, we found more SNPs pointing to a locus incorporating many genes of the UGT2B family and the UGT1A family. And for the metabolic ratio, we again found a locus pointing to genes of the UGT2B family and the CYP2C family. What we saw from looking at these loci individually was that they colocalized with loci that came out of GWASs of other traits, particularly traits related to coffee metabolism, so for example, caffeine plasma concentrations, and also, interestingly, with a GWAS that had been conducted on patients. Pelargonate, you might not know about, is a fatty acid, but it's structurally very similar to valproic acid, which, as you might know, is a drug that might have interactions with Clozapine. So we thought this was interesting, and it was very reassuring that the first time we've done a GWAS of metabolism concentrations, which I can tell you technically is very different to doing a GWAS of, for example, schizophrenia, we found this sort of results. We've recently had done a GWAS of an expanded sample of CLOZUK that now incorporates 16,000 assays for under 4,500 individuals. And for the first time, we've been able to analyze not only the individuals of European ancestry in CLOZUK, but also individuals of African and Asian ancestry. All of the CLOZUK samples were recruited from the UK. So as you can imagine, these individuals of non-European ancestry form a minority of our samples. Because they were a minority, we had to exclude them from quality control purposes in previous studies. But now our techniques and our way of looking at the data has matured enough that we can include them again without any worries of confounding our results. We've run 3 GWASs again. We found many of the loci, and reassuringly, we now start to find convergences. We find that the UGT1A variants are also a locus for clozapine, as they are for clozapine. The 1A1, 1A2 variant is a locus for clozapine, as it is for clozapine. And we even find a new locus, which is POR, which is a regulator of the activity precisely of the 1A2 enzyme. So everything seems to be fitting quite well together. I also want to highlight, although I'll go into it a little bit more later, is that these markers, these genetic markers, and derived polygenic scores that we calculated from these markers, we found them to have effects in both the European and the non-European ancestry. So a very interesting result that we got from finally looking at a diverse data set. So, as I said, all of these results within the reassuring, and they're reassuring because they map quite neatly to genes that appear in the clozapine pathway and that we know they're important for clozapine metabolism. We have your 1A2s, we have your 2C19s, and here we have the UGT enzymes. All of these genes now have genetic variants with effect on metabolism that we have mapped through the GWASs. Of course, many of these are pending replication, others have been replicated by other studies, and I'll go into that now. So how far or how close are we to something that we could use in clozapine management? This is always very difficult to say, but to give you an idea of what the effects of these genetic variants translate to, I just go to the intergenic 1A1, 1A2 variant, which being intergenic is the reason why this variant wasn't discovered before, because no one was typing it. It had to come a genome-wide study in order to find it. This variant is known to affect the regulation of the genes 1A1 and 1A2. And the effect that we find, this is a variant that is in the UK has a 24% allele frequency, is equivalent to reducing the clozapine dose in 50 milligrams a day. Collaborators that we have in the University of Oslo have found similar results. They express them in a different way, 15 to 18% reduction in clozapine plasma levels, but also they did have a smoking behavior in their sample, and they saw that those reductions were independent of smoking behavior. They also found another variant in their analysis in a gene called NFYB, which we haven't been able to replicate or to even look at because we don't have data, or arrays and our imputation doesn't have any data from that region. It's also a 1A2 regulator. It has additive effects. So they've concluded in a very recent study that the smokers with both minor alleles might need 3 times the clozapine dose compared to non-smokers to reach the therapeutic plasma concentrations of clozapine. So this is the sort of significance of the effects that we're talking about. And again, this needs to be investigated in more detail, but this is where we're starting at. Even though all of this research is being conducted, we're already getting an idea that clozapine metabolism is likely an oligogenic trait, even if we didn't have that diagram of the clozapine pathway. What the genetic data is telling us is that it's a tiny fraction of variants in the genome that are going to have effects for it. And the variance explained by these genetic variants, what we would call the heritability normally, is not massive. It's about 3% for clozapine and 5% for non-clozapine. But then, if we derive polygenic scores using only the genome-wide significant variants, and again, these are a handful of variants in a handful of genes, we can explain between 20 and 30% of that heritability. So that means that our variants are having very large effects, and because they're very few, this should make it an easier replication. It should be easier for studies to conduct, to look at these variants in detail when they also have clozapine samples, or even for trials to be conducted using just this information. So I just want to finish just a few slides before I finish with the idea that having this genetic information is not going to be a a tool on its own that can condition or greatly determine the treatment pathway that an individual is going to follow. But as the International Society for Psychiatric Genetics says, genetic information needs to be a decision support tool, something that is incorporated to other tools, such as TDM, such as clinical guidelines, things that we already have. And in this plot, those would be the pipes in white and the pipes in blue, and the genetic information will be the pipes in green. The genetic information needs to be incorporated to our existing pathways and needs to enhance them, not substitute them. And as the paper that put this figure forward says, and I think it's a nice way of phrasing it, we need to have a distinction between things that might be nice to know and things that we need to know, because the statistical significance of the result does not tell us much about its clinical relevance. So in order to be able to know whether we need to know about these genetic variants, these pharmacogenetic variants, we need research that tells us whether the individuals that have those variants are at greater risk of non-response, of adverse reactions, or of discontinuation. We need outcome data. And ideally, we need, at some point, prospective trials. And I know that there are European initiatives that are going in this way, and I know there's a UK initiative, fortunately, as well, on antidepressants to look at whether prospective trials can help us and how they can help us. The situation can be even a bit more complex than what I've highlighted here. And this is why we need to have the door open to doing lots of research with different outcome data, because it's not necessarily going to be all pharmacogenomic variants related to metabolism. We have done analysis where we have shown that clozapine doses might have a genetic component as well, but that genetic component is not related necessarily to clozapine metabolism. So it's been thought for a long time that this delicate balance at adjusting clozapine doses or doses of any drug is related, is this balance between tolerability and response is very, very much determined by metabolism, by genetics that have to do with metabolism. But when we look at a CLOZUK sample, and also in samples from our Norwegian colleagues, at the polygenic scores that from the upper 2.5% of the individuals, so individuals at a great genetic risk of schizophrenia, if you allow me to say it in this way, and we looked at individuals that had high doses of clozapine, over 600 milligrams a day, what we found is that individuals at a high genetic risk of schizophrenia were more likely to have these high doses. And once you go to the extremes of genetic risk of schizophrenia, the high doses of clozapine were nearly twice as likely than in the other groups of individuals. So the genetic variants that are related to risk of schizophrenia, not to clozapine metabolism, might be having effects, for example, on severity of the disorder. And that severity of the disorder, of course, might condition the antipsychotic dose that individuals need to receive. Though, as we say on this paper, it's not entirely clear that those processes are not entirely clear because we don't have response data, but this is a hypothesis that we put forward. As well, when we looked at adverse drug reactions, and we did this study a few years ago led by my colleague Sophie Legg, when we look at clozapine-associated neutropenia in a subset of CLOZUK of African ancestry, we found a genetic variant which is fairly exclusively found in individuals of African ancestry, is nearly exclusively found in the African continent, which had very strong effects. And the reason why this variant has very strong effects for clozapine-associated neutropenia is because it has very strong effect on the neutrophil concentrations in plasma. 97% of carriers of this variant had been diagnosed via the normal hematological assessment route with benign ethnic neutropenia. So they had been diagnosed with neutrophil levels that were constitutionally, not pathologically, lower than the rest of the population. And we found a genetic marker for that with very good predictive value. Which also allows me to highlight that if I'm calling for conducting more studies on clozapine, more studies on treatment outcome, even prospective trials, I also need to highlight that we need surely to be including everyone in our genomic studies. The picture that you have here on the left is the diversity of participants in all genome-wide association studies that have been published, not 5 years ago, yesterday. About 95% of the individuals that have been reported in genome-wide association studies are of European ancestry. This doesn't match the world diversity at all. And when we look in our work on clozapine, we know already from the guidelines that individuals of Asian ancestry are more likely slow metabolizers, they get high clozapine levels. What we find in other studies is that individuals of sub-Saharan African ancestry are likely, at least what we have seen so far, fast metabolizers, and they get constitutionally low levels. So that's also going to affect the dosing. And so far, that's not reflected in any guidelines. We think we're the first ones to report it. So of course, it needs replication, it needs more investigation, but this is the situation that it is. So I just want to finish with my existential questions slide. What do we do with all of this? If you allow me to be bold and to speak from the point of view that we have in the Cardiff group, we need to do pharmacogenomics research, adopting some of the pipelines that have been trialed and been proven as true in the complex disease genomics world. We need large scale and particularly collaborative discovery. We need to share samples. We need to find frameworks to securely share samples, securely share data. We need to favor independent replication, even if that needs pressing funders for funding studies that are mainly going to be for replication. And we need that once a genetic variant is shown as promising, we rapidly adopt it in follow-up studies that might have different study designs, that might have been looking at different things. And here I highlight 2 papers, that one that has that data on smoking that we didn't have in our analysis, and another one which look at interactions between clozapine and valproate. They looked at the genetic variants that had came from our GWAS test. They look at whether they have effects. And this is the way to go, adopting those results quickly, showing whether they are useful for doing other things. If any of you are interested in genotyping and are interested in pharmacogenomics, there is research that shows that if you have array data, you can call star alleles as well as if you have the whole genome sequencing. And I haven't gone into star alleles for clozapine because we haven't still called them. It's technically complex with the data that we have, but that's a work in progress. And just to reinforce that, we need to make all this data publicly, at least through publication, because pharmacogenomic guidelines, expert international consortia, all of it, they require the data to be publicly available. They won't review private data. So thank you, all of you, for listening to me and for inviting me again. Just want to highlight my excellent colleagues who have helped with many parts of this research, not only Jennifer, Sophie, and Isabella, my former PhD student. I also want to highlight Millie, Lisa, and Francesca, who helped with the last clozapine GWAS, and of course, James, Mick, and Mike. Without them, none of this, and without our funders and our collaborators, none of this will be possible. So thank you very much, and I am open to any questions that you might have.
Yeah, Antonio, thank you very much for the fascinating talk and the complexity of what you have been done, which is extraordinary. Thank you very much.